In this lecture, we're going to talk now about mitosis. Now, mitosis is an absolutely essential process for life. It is a type of cell division that produces two exact copies. So that mother cell is going to divide into two daughter cells, each of which is identical to that mother cell and each of which is identical to each other. Now, this process is absolutely essential for things like reproduction, growth, and development. So you start off as a single-celled embryo, and of course you are more than just one cell as an adult, and so to get more cells, that single-celled embryo divides by mitosis. And mitosis is required for tissue renewal. When you damage some skin cells, for example, you need to divide more cells to make more and replace those skin cells. The mitotic phase is one of the two phases of the cell cycle. The first phase being interphase, and we're going to discuss interphase in some more detail a little bit later, G1, S, G2, and it's the S phase in which DNA replication occurs, which we have just talked about. And then the other phase of interphase is the mitotic phase. Now the mitotic phase has 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. If we take a look at a chromosome during mitosis, we have to make sure that we come to an understanding of nomenclature, of naming, of terminology. Because if you don't understand how we name things, then when you read stuff in the book, when you read stuff on an exam, it's going to become very confusing. Now, 99.9% of the time, a chromosome, right, is just one double-stranded DNA molecule. Okay, it's millions of base pairs long, it has thousands of genes, and it just exists like this, wound around various histone proteins and being the substrate for transcription. But if that cell is going to divide, what happens during the S phase of interphase is this double-stranded DNA molecule duplicates. So now you have 2 of them. These guys are 100% identical to each other. This isn't a case where one of these comes from mom and one of these comes from dad. These 2 double-stranded DNA molecules are 100% identical to each other, and they are connected to one another by a centromere. Okay, that's this constriction here that is connecting one of the double-stranded DNA molecules to the other double-stranded DNA molecules. And in this particular situation, we call them sister chromatids. Now, as long as sister chromatids are connected by a centromere, like we see in this figure here on the left, we still consider it one chromosome. Now, it is a duplicated chromosome. It consists of 2 sister chromatids, but nevertheless, as long as it's connected by a centromere, these are considered, this whole thing is considered one chromosome. Now, as soon as the 2 sister chromatids separate as a result of mitosis, then each one of the sister chromatids, since they are no longer connected to one another by a centromere, they are now considered 2 separate chromosomes. Okay, so make sure that you understand the terminology here. Now, you are going to have lots of practice differentiating the various stages of mitosis in lab. The book has a very nice figure on it, but I'll quickly run through them and help you out, and we'll talk a little bit more detail about things like the mitotic spindle as we go. Now, interphase is not part of the mitotic phase. Interphase is one of the 2 phases of the cell cycle. Okay, but we're going to start here anyway. And remember, a cell is in interphase 99.999% of the time. It's only when that cell is going to divide when it goes through the mitotic phase of the cell cycle. It is during interphase in which the centrosome duplicates. Okay, that's going to go eventually to set up the mitotic spindle. The nuclear envelope is intact. The DNA is decondensed, so you will not be able to see individual chromosomes. And you'll be able to see a dark spot within the nucleus. That's called the nucleolus, and that is where ribosomal RNA is made and where ribosome subunits are assembled. The first phase, the first stage of the mitotic phase of the cell cycle is called prophase. In prophase, the two centrosomes begin to move to opposite sides of the pole. The nuclear envelope is still intact, so you're still going to see those chromosomes being confined to a very specific region within the cell. And the chromosomes then begin to decondense. Now, prometaphase, the difference between prometaphase and metaphase is pretty subtle. The most important thing that you can see under a light microscope to differentiate the two is the breaking down of the nuclear envelope such that the chromosomes aren't confined to that perfect circle anymore. Now, what also is occurring is they are condensing, the chromosomes are condensing a little bit more and beginning to connect to the mitotic spindle. And we'll talk about what the mitotic spindle is in just a few minutes. Finally, those chromosomes are finished being connected to the mitotic spindle. They line up along the metaphase plate, and this is the phase that we call metaphase. This is one of the easier phases to observe under a light microscope. This is also the longest phase of mitosis as these chromosomes continue to try to organize themselves on that metaphase plate and connect to the mitotic spindles. Now, I've mentioned mitotic spindle a couple times, and let's take a look at exactly what that is. So the centrosomes here that have been duplicated during the interphase of the cell cycle have gone to opposite poles of the cell, and they extend three types of microtubules. The first type are aster microtubules, and those aster microtubules are what sort of anchor the centrosome to the pole of the cell. There are kinetochore microtubules, and those extend and attach to the kinetochores of the chromosomes. Now, the kinetochore is protein complex that binds to specific regions of the chromosomes, and these kinetochores then also bind to kinetochore microtubules. This is how those chromosomes are going to be aligned on that metaphase plate through actions of the kinetochore microtubules interacting with the kinetochores of the chromosomes. And the third part are called non-kinetochore microtubules. These extend into the metaphase plate, but they do not bind to chromosomes. Instead, they overlap one another, and they help separate the two cells, the one cell, the splitting of the one cell into two. Anaphase is next. Anaphase is the phase at which the sister chromatids that have been linked together by a protein called cohesin. Well, at this point, that protein, and we're going to talk a little bit more about this protein later, is split, and that allows the sister chromatids to separate. So now, at this point in this image, we have 1, 2, 3, 4, 5, 6 chromosomes. Now, they are 6 duplicated chromosomes, but they're 6 chromosomes nonetheless. As soon as those sister chromatids separate, now we call each one of them a unique chromosome. So now, at this particular point in time, we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. Now, the way in which those chromosomes, the sister chromatids separate and the sister chromatids move to the opposite poles is through the kinetochore, this protein complex that binds to the centromere of the chromosome. And it contains a motor protein that interacts with microtubules. So these are things that you learned in chapter 6 so long ago. And that motor protein uses ATP to move along that microtubule, dragging that chromosome along with it. So this is the way in which we get this motion, this movement of sister chromosomes to the opposite poles of the cell. Eventually, those sister chromatids will move all the way to the opposite poles of the cell, and then we get a reversal of everything that we started with, which means that the chromosomes start to decondense again, which means that the nucleolus begins to reappear. The nuclear envelope begins to form again. And finally, we get a process referred to as cytokinesis. Cytokinesis is the process by which the cell membrane splits into two. Technically, mitosis is the division of the chromosomes, and cytokinesis is the division of the cell membrane. Now, in animal cells that lack a cell wall, what the cytokinesis involves is something called a contractile ring of actin microfilaments that contract to form this cleavage furrow and eventually pinch all the way off to form the two daughter cells. In plant cells that have the cell wall, a cleavage furrow is impossible. So instead, what happens is you get these little vesicles forming that separate one cell from the other cell that's going to eventually emerge. And these vesicles then fuse to form what's called the cell plate, and then that cell plate grows to separate the two cells from one another. Okay. So in lab, you are going to be looking and trying to find in the microscope vesicles and the cell plate of onion root tip cells. All right. So that concludes the various stages of mitosis. And what the next lecture is going to involve is understanding how the process of mitosis is regulated and how the cell cycle is regulated.